Okay, great. So when I was thinking about what I would talk about today, I sort of, I've developed a bunch of methods in the last couple of years, one including with Bianca, to think about how to do imputation in single cell RNA sequencing data. But I sort of was thinking about more broadly how I've used imputation for missing data in the past. And I wanted to share with you some experiences that I've had. And this is sort of for general biomedical data. I'll go through a handful of data sets where I've done this and had imputed data in my research. So first of all, for biomedical data, missing data are everywhere, absolutely everywhere, sort of expected. So when we think about genotyping, when we think about finding phenotypes, I'll give you some examples. RNA sequencing has this sort of dropout phenomenon, which we'll talk about. Even if we're talking about medical data where you're looking at a patient in a hospital, a lot of data are basically always missing. In my experience with the biomedical data that we're looking at, the data are really never missing at random. There's always some structure, some component of the missingness that is drawn from the sample itself. When I think about the studies where we've done missing data and imputation of that missing data, sometimes these downstream methods require complete data. So we need to impute those data. If you think about like a neural network, which really requires you to have a complete data set, not allow you to have NAs, that happens. Sometimes imputation is good enough to identify signal. Often we just do mean imputation. I'll give you some examples where actually mean imputation or the sort of sample and hold ideas work pretty well. Very, very simple. Sometimes researchers just sort of cross their fingers and hope that imputation is good enough, but they really fail to calibrate for the specific example that they have. This happens a lot with genotype imputation, where the imputation methods calibrate during the analysis of the method itself, but when you actually apply it, it's not entirely well calibrated on the data set. And I think important, yeah.
I'm sorry, I think there's a question from the audience.
Oh, yeah.
Andrew, do you have a question? Oh, I think he had his hand raised. I'm sorry.
Okay, hold on. Sorry, kid walked in. Okay, yes, good. Okay, so let me know if there's another question. Okay, thank you. Okay, and then I think importantly too, uncertainty in the imputed values is often not carried forward into the downstream analyses that happen. Okay, so starting off with DNA genotype imputation, which is probably something that people have heard about. In these genome-wide association studies that we perform, often we need to correlate, or the goal of the studies is to correlate genotypes with a specific phenotype of interest, so we can find the underlying genetic basis of important phenotypes like disease. And here's a picture from the Impute2 website, which I think is sort of informative in this. We have some sort of very dense reference genotypes. So let's just assume we're dealing with 0 1s, meaning 0 is 0 copies of the mutation, 1 meaning 1 copy of the mutation. Each row here is going to be an individual. Each column is a particular mutation in the genome. And we know from biology that there's going to be very nice correlation, often locally, among the values of the genotypes. And so the goal for imputation here in this missing data set is that here's our study data below. And we note that there's a lot of columns where a lot of SNPs that just aren't sampled. And so you can imagine a chip, a genotyping array, where you only have a handful of this dense matrix that we have above sampled on the array. And the idea then is to use the correlation that we know about within a population to be able to impute all those question marks using our reference sample. So that's really the goal with genotype imputation. Sorry about this funny figure. This is all I had for this. But for single cell RNA sequencing, the idea is that often one of the downstream goals that we have is inferring the cell type. So the idea there is that for each cell that we've sequenced the RNA for within that single disassociated cell, we'd like to figure out what cell type that cell is actually of. So we can figure out, for example, the proportion of immune cells floating around in the brain, or the proportion of neurons in the brain, and all these questions. And so what we get, though, is actually a count of the number of fragments of RNA that map to particular genes. And when we only sequence a handful of those fragments, even if we sequence 10,000 or 100,000 fragments, there are a lot of genes, RNA, that's actually not represented well in that cell that simply won't be sampled because this is a multinomial sample. And so you see all this blue here represents basically zeros in the data. So whereas actually the cells and the genes here in this matrix should be fairly dense because there's usually a low proportion of RNA in each of the cells, we get a lot of zeros just because we can't sample every single piece of RNA in every single cell. And so the idea is for imputation then, not even for imputation, the idea is that even if just looking at these zeros, we see that the more frequent a particular gene is, unsurprisingly, in a cell, the more likely it is to have a non-zero value. So to say it the other way, if it's a lower abundance RNA, it's more likely to drop out, just according to this multinomial frequency. Another way that we have missing data is looking at longitudinal studies and phenotypes more generally. So again, when we think about association studies, our goal is to identify associations between genotype and phenotype, but then also covariates too. If we have phenotypes and important covariates like race or age, there's often going to be missing data due to dropout, where dropout here represents, an individual dropping out of a particular study. There's often a situation where even though the individual hasn't dropped out of the study, they've just failed to follow up for one reason or another. There's also non-compliance, where a patient or someone in a study says they will follow a particular intervention like exercise or diet, and they fail to do that, and it's hard to get the appropriate phenotyping data from them. So some samples of this that I've worked with are the Fragile Families Childhood Wellbeing Study, which we're going to talk about in a little bit, and also the UK Biobank. UK Biobank, for example, has 500,000 people, and really at this point, they're only followed up 7 to 10 years after they were initially looked at if they end up in the hospital, which is not a great reason to be followed up. It means they're generally more sick, so the healthy people aren't followed up with. Here's another example of data that I've played with that need to be imputed, that there's a lot of missing data, is hospital patient data. So we have about 270,000 patients from the UPenn hospital system. This is a single patient over about 50 days of observing this patient in the hospital, and you see time series data. But you see huge regions for systolic blood pressure, for example, huge empty swaths of data. And this generally represents, a time where the patient is maybe being moved from rooms, maybe just the nurses or the clinicians failed to enter the data on their chart. You also see sort of randomly scattered data that are only sampled sporadically, intermittently. All of these mean that if we want to find the exact state of a patient at a particular time, we need to do something a little bit more clever to be able to try and figure out what's going on with a patient right now, or maybe even slightly in the future. So a handful of common methods that I've used and followed through with in missing data sets for biomedical data are the following. So first, as was discussed earlier by Julie, impute the population or to the feature mean or median. So this is a very straightforward thing to do. For a particular feature, you're going to compute the mean or the median of the non-missing data, and you're just going to set all of the missing values to that mean or median value. Very simple, but also obviously, as mentioned, really changes the feature distribution sometimes, and is often just not representative at all. No variation is included in there, so it's very, very naive, but sometimes it actually works quite well. In medical data, as I said, there's this idea of sample and hold, which is what a lot of methods do, sort of surprisingly, where you basically take the most previous time point, the most previous observation of a lab, say, white blood cell count, and you just assume that until it's sampled next, it's going to keep that exact same value, even if it's hours or days later. It's just going to maintain the exact same value. Many methods, including probabilistic PCA, are based on dimension reduction type methods, and we have some methods in that. That's one of the things I developed with Bianca, for example. And the idea there is basically that you have an observation matrix that has missing data. And so you use EM, and this is again, as Julie said, you use some version of expectation maximization to first sort of guess the values of those missing data set. Build the dimension reduction, and then iterate, reconstruct the missing values using that dimension reduction, and continue to iterate until you have a reasonable set of values for the missing data in the original data set. And probabilistic PCA is one example, but there's many examples of this. Another approach is iterative imputation, again, as Julie mentioned, where you basically build a classifier for each feature, conditioning on all the other features or even some subset of features. So this classifier can be completely arbitrary. You can use linear regression, you can use random forest, you can use neural network. And then again, iteratively, you continue to re-estimate all of the missing values conditioned on everything else as you sort of, again, iteratively fill in all of the missing data in the remaining features. Genotype imputation relies on essentially a version of a hidden Markov model, and relies heavily on, again, linkage disequilibrium, which is this correlation among neighboring SNPs. So given a covariance matrix, essentially you can describe the imputed values for those missing variants. And then finally, something that Don Rubin wrote about in 1987 is this idea of multiple imputation, where you basically take your missing data matrix, you impute the data m different times, and the idea is essentially to capture uncertainty in the data matrix itself. And so the idea is that then when you do downstream analysis with these m different imputed data matrices, you're going to get some idea of uncertainty propagated into the downstream results. So I'll tell you that all of these methods that I've used in practice have real drawbacks. As much as they're theoretically very nice, there are some pretty substantial drawbacks to each of them. So for example, when I was a postdoc many years ago with Matthew Stevens, we have two different studies, genotyping association studies, where in the first one, we actually look at the associations of a particular mutation. This is the ID number of that mutation. And here's 2 Gs, which is the non-mutated allele. And here's one version of the mutated allele, an A and a G. And here's 2 versions of the mutated allele, AA. So for these different 3 genotypes, we see that actually the values of gene expression of this particular gene, GATM, decreases in a very nice linear way. There are obviously big old error bars there, but the idea that is hidden in this graph is that actually we imputed some of these values of the genotype. I'm sorry, I need to close my door. Okay, sorry. I'm back. Okay. Here's a second one. Oh yeah, so what's being hidden here, I should say, is that we imputed a lot of these genotypes. So there isn't actually all 0, 1s, and 2s. Sometimes there are values in between, which represents the median, the mean value, expected value of the number of rare variants. So the minor variants. Similarly here, we did imputation on these, again, hidden in this graph, but this is association with the euphoric effects on about 200 undergrad students at University of Chicago of amphetamines. And we see, again, a really nice association of these different genotypes with the trait that we have here. But again, we're missing the values of imputation. We're sort of locking them at 0, 1, or 2. And so when we actually published these studies, one thing that was necessary to do was to perform imputation on the genotype values and then do these associations. We did that, but then we actually genotyped these missing values because we have the original samples. And what we found was that actually the genotypes didn't match the imputation very well at all. So the genotyping methods, unfortunately, were not appropriately calibrated in terms of their uncertainty. In the end, when we actually looked at the sort of resulting associations, the strength of the association was pretty substantially reduced after doing the genotyping, which basically means that even though, again, these methods were originally calibrated very nicely on sort of existing data, on this sort of real world wild type data, wild type in the sense that it's not training the models themselves, we see that they're not well calibrated at all. Here's a second thing that I did with imputation. So this isn't the one I developed with Bianca, but it's another method that we developed in my group for imputation of genotype or gene expression. And the idea is basically to use a GPLVM, and then we added actually a heavy-tailed residual student's t-distribution to allow for a lot of zeros, which ended up often messing up the imputed manifold that we developed for this. And so we performed this type of analysis, this dimension reduction for imputation in a lot of different data sets. Here's an example of immune cells using 10x sequencing. And here's dimension reduction using all 20,000 genes, but then we only overlay the values of a single gene. This is a tyrosine kinase binding protein. And you can see, again, a lot of zeros in these data representing the sort of dropout. But you can see that, well, one thing that's nice to look at here is that, first of all, there is sort of a gradient, even though with this particular gene expression levels, even though these methods were performed on 20,000 genes, and it is robust to zeros. So it looks like a nice sort of clean result. But I guess sort of the punchline of this is it would be really nice maybe if these methods, for example, cell type imputation methods, were robust to a lot of these zeros, didn't rely on, for example, single markers to be able to determine cell type. Similarly here, we're imputing the trajectories of these 409 T cells across different days of infection, so 0 to 7 days post-infection. And again, here we see that there's a lot of sort of magnifying and weirdness going on in the tree trajectories because of the zeros. However, again, this was a approach that was fairly robust to the number of zeros in the data set, to the missing data. And so it ended up doing a fairly nice job of this reconstruction overall. The sort of another depressing example I have of this is the Fragile Families Challenge. So I know I'm sort of running out of time, but I'll try and be very brief. This is an absolutely beautiful data set with 5,000 children that were born to single mothers, usually on welfare. And the idea is that across 15 years, they sample and have survey data for each of these children. The challenge itself took the background, so it's almost 13,000 variables from birth to age 9 for these 5,000 kids. And then the training data set also included about half of the data for age 15 and 6 of the outcomes that I'll show you. And then the holdout data is also these 6 outcomes at age 15. The idea is just to predict these data with as high accuracy as possible, predict the holdout and the leaderboard data with as high accuracy as possible. So the Fragile Families Challenge, they wrote up this in this 2020 paper in PNAS recently that I was a part of, but hidden in there somewhere. And basically showed that imputation with social science data is incredibly challenging. Not surprisingly to anyone, I think, but how bad it was really challenging. So the challenge itself included on the order of 200 different approaches, methods to be able to try and impute material hardship, grade point average, grit, whether a family was evicted, the results of job training, and whether a family member was laid off during this period when the child was at age 15. And what these graphs are showing is essentially, as opposed to benchmark, which is, you know, on the order of sort of mean averaging, it's very, very simple, very simple imputation. The best submission was only sort of epsilon better in most cases, and in some cases not at all, like the job training data. So this is kind of scary, but also in my mind sort of talks about how challenging imputation really is, because all of this is really just the act of imputation. The fourth example that I wanted to talk about really briefly is the patient data, where again, we use these beautiful Gaussian processes. And we have, they're challenging to fit, but we use multi-output Gaussian processes to try and do imputation along each of these patients. But again, when we actually used those imputed data for reinforcement learning later on, where the reinforcement learning included all of these state variables which had been imputed, we found that actually the policies that we estimated didn't perform any better when we used our fancy imputation with a multi-output Gaussian process, as opposed to using just the sample and hold that a lot of other groups did. So this was kind of frustrating, but also, I guess, understandable. So I just wanted to conclude briefly by saying missing data, I think, are very hard to work with, but also ubiquitous in biomedical data. The more sophisticated methods sometimes don't necessarily improve the downstream results. For the not missing at random data, which is sort of the de facto reality for all of the data sets that we're looking at, preserving the feature distribution may actually not be the right thing to do, simply because the feature distribution reflects the biases in the data collection. The methods are really hard to calibrate. I think there's a lot of sort of echo chamber problems with calibration of the imputation methods. Often we find that the imputed values are not within range. This happened a lot with our medical data in particular, which made it really hard to work with sometimes. Unlike Rubin's method, which we actually never used, but I wanted to throw it out there as something that we could use, uncertainty is not well propagated to downstream methods appropriately. So we really don't know in a lot of cases how uncertainty in imputation is reflected in our results. And so I guess sort of to put it all together, when actually working with these methods on biomedical data, I highly advise you to think about calibrating uncertainty within your own data, check the ranges carefully, actually observe. So if you have missing data, go back to the original data set and see if you can observe those values before you make any serious claims about your results based on imputed data. And here's my amazing group and a bunch of collaborators and some funding sources. Thank you.
Thank you so much, Barbara. So there are a couple of questions for you. So one of them is there any one of these critical issues with biomedical data that is the most important to solve at this time? What is such issue? And are there any most ripe research areas that would require broad solutions that you could talk about?
Yeah, both good questions. So Yeah, so I think most important things right now, I mean, I guess I would say thinking about methods for um, downstream analysis that allowed missing data. So a lot of the most powerful methods right now require complete data sets. And I'm wondering if instead of throwing complete data sets that have been imputed maybe incorrectly, maybe we build these downstream methods to allow data that are have missing features in them. So again, a lot of deep methods right now require full data sets, and it would be really nice to think about either different approaches or different ways to actually sort of the Bayesian approaches where you can sort of not include any data that's actually missing, not requiring the missing data to be imputed, and actually get those downstream results. And specific areas, sort of application areas for missing data. I mean, I guess theoretically, whether it's causal inference or association, I think a real question is how to, again, incorporate missing data in these downstream examples without losing statistical power. So, going back to the UK Biobank data set, it's great to have 500,000 people, but if we don't have follow-up data about heart attacks for a third of them, then we simply can't say that they're healthy, that they're not on statins. And I think, again, using data points inappropriately, using samples inappropriately, as opposed to taking that a slight reduction in power, I think we need a little bit more work in understanding that, whether it's causal inference or association.
Thanks. There are two more questions. So the previous question was from Laura Balzano. There's another question from George Linderman. In these examples, such as fragile families, which show that imputation is hard, do you think it is a limitation of methods or that the features are not so predictive? What is it that makes imputation hard in that particular setup?
Yeah, that's great. I think it's the whole structure of the problem, but I actually, so for that, there were actually basically 200 different methods applied to each of those 6 different phenotypes. So I don't think it's an issue of the method not being good enough. The methods were quite good. I mean, they were all of the methods, so it's hard. We just threw everything against the wall to see what stuck, and nothing stuck. So they were very sophisticated, sometimes hand-tuned methods, essentially, to just deal with each of these different 6 covariates, and nothing really worked. Oops, wait, where is it? And nothing really worked. Again, like for GPA, the best submission is only slightly better than this sort of baseline GPA imputation. So this is quite scary and disappointing. So I do think that this is probably an issue with the structure of the supporting covariates not being very predictive, not being signal there. That said, there are some really interesting parts of imputation that I think we can learn from this example, but a lot of issues. Of course, missing data is part of it, but also very biased zeros and ones in the case of layoff and eviction, for example, and job training, make it incredibly challenging to perform these analyses generally. But I think there's very little signal there. Yeah.
And the final question from Andrew Zhang is, just out of curiosity, what's your goal of using reinforcement learning for the imputed data? And in what principle the performance is good or bad? How do you evaluate that?
Yeah, great. So in this case, for this particular example, actually, this is unpublished. I can tell you some other studies, but this is for thinking about when to replete electrolytes in a patient in the ICU. So when a patient's in the ICU, they often lose different electrolytes very fast. They have to essentially be given IVs or oral, basically Gatorade, electrolyte replacement therapies. And when we're thinking about building a reinforcement learning policy to describe when and how much and the method by which each patient gets these electrolytes, we have to look at all of these different variables, again, which may not have recent existing values for the particular patient. So we actually ended up sampling them every 6 hours for electrolyte repletion. And again, the policies were driven by observing these different covariates. So for example, heart rate was looked at slightly for determining this, but if you have a higher heart rate, you're more likely to get electrolytes, for example. So sort of putting it all together, the idea is then that, the evaluation is then figuring out whether patients stayed within range of particular electrolytes throughout their stay, and whether or how many times electrolytes were repleted. So there was a cost measure to it as well. And we didn't see major differences between doing imputation using the sample and hold, the very simple methodologies, rather than doing more sophisticated methodologies. So they sort of evaluated about the same, and the policies were nearly identical.
Thank you very much.
Thank you.
Laura, do you have, I think we have a couple of comments as we go forward. Thank you so much, Barbara. Great talk.
Yes, thank you very much.